Tonight, somewhere on the salt flats of South America, in pools so salty almost nothing else can live, the water is faintly purple. It is the color of a microbe, a single-celled survivor of an older world. And for nearly 2 billion years, that color was not a curiosity in a salt pond. It was the color of the earth. There were no trees, there were no flowers, there was no green anywhere. The continents were bare gray rock, scoured by wind and weather. The sky, depending on which century you stood in, was hazy orange, or thinly milky, or pale and watery, but it was almost certainly never blue in the modern way. The oceans were dark and shallow and warm. They smelled of iron and sulfur. They were full of tiny living things that we would not recognize as plants, and that did not look like anything alive on the surface of the earth today. And yet, somehow, those tiny things had learned the most important trick that any creature on earth has ever learned. They had learned to drink the light, to take the energy of the sun and turn it into the energy of life. They had invented photosynthesis, or rather, a kind of photosynthesis, a simpler kind, one that used a different molecule, in a different color, in a different chemistry. And the molecule they used has not vanished. It is still around. It is in the bacteria living on those salt flats today. And in a small but very real way, it is also inside your own eyes. This is the story of the color of life before chlorophyll. The story of the time, 3.5 billion years ago, when our planet may have looked almost lavender from space. The story of how green eventually took over. And the story of what little is left today of the world that used to be. So settle in. Let your shoulders ease. We are going to travel slowly tonight, the way light itself travels through deep, still water. Back through the long quiet of the Archean, to a younger sun, a younger Earth. To understand why some scientists now think the Earth was once purple, it helps at first to understand the color of life today. When you look at a forest, you are looking at chlorophyll. Chlorophyll is a green molecule. It sits inside the cells of every leaf, of every blade of grass, of every vine and every moss. It also sits inside vast numbers of microscopic plants in the ocean, the photosynthetic plankton that drift in the upper 100 meters of the sea. Chlorophyll is, in fact, one of the most successful single molecules in the entire history of the Earth. Wherever there is sunlight and water and life, there is chlorophyll, or something very like it. And chlorophyll is green for a strange reason. It is green not because it absorbs green light, but because it does not. Chlorophyll absorbs red light. It absorbs blue light. It uses both to power the chemical machinery that turns carbon dioxide and water into sugar. But the energy-rich middle of the visible spectrum, the band of green and yellow that the sun sends to us most strongly of all, chlorophyll mostly ignores. It bounces that part back into your eyes. That is why grass is green. That is why the entire biosphere of the modern Earth, when seen from orbit, has the color of the sun's leftovers. This is, when you stop to think about it, a peculiar arrangement. The sun is brightest in the green-yellow part of the spectrum. It would seem that any photosynthetic life form that wanted to be efficient should be using exactly that bright peak, not bouncing it away unused, and yet chlorophyll does bounce it away. The most powerful part of the sun's light, the part our atmosphere lets through most freely, is the very part that almost all of the modern biosphere refuses to drink. For a long time, biologists treated this as a quirk, a historical accident. Chlorophyll worked. Life used it, and the rest of the spectrum was, evolutionarily speaking, no longer available. But starting in the early 2000s, a small group of scientists began to wonder whether the gap had a story behind it. whether it might be the trace of an older photosynthesis, something that came first in deep time and used the bright middle of the spectrum so thoroughly that any later kind of photosynthesis had to work around it. That older photosynthesis, the one some now believe existed first, did not use chlorophyll. It used a much simpler molecule, a molecule with a long, bright history of its own. A molecule, in fact, that you carry with you everywhere is called retinal. It is a small organic compound, only 20 carbon atoms long, with a chain of alternating single and double bonds. That structure is unusual. It is what allows retinal to bend like a tiny spring when a photon of light strikes it. The energy of the photon is enough to twist the molecule into a different shape, and that twist is what living cells of certain kinds learn to harness. The most famous use of retinal today is the one inside you. Inside your eyes, in the rod and cone cells of your retina, retinal sits embedded in proteins called opsins. When a photon of light enters your eye and strikes one of those retinal molecules, the molecule bends. That bending triggers a tiny chemical signal which travels down the optic nerve and ends as a glimmer of perception inside your brain. Every star you have ever seen, every face, every word on a page, every cloud, every passing animal in the corner of your vision came to you because a tiny retinal molecule somewhere in the back of your eye changed shape under the touch of light. But retinal does something else as well in other places. In a particular group of microbes that today live mostly in the saltiest places on Earth, retinal is not used to perceive light, it is used to drink it. Those microbes are called Archean. They are not bacteria exactly. They belong to a separate, very ancient branch of the tree of life called the Archaea, single-celled organisms whose biology is, in some ways, even older than that of bacteria. And in their cell membranes, Archaea carry a protein called bacteriorhodopsin, which carries retinal in its structure, and which uses the energy of light to pump protons across the membrane. That sounds technical, but it is, in fact, the simplest known way that any living thing can capture energy from the sun. Light enters, the retinal bends. The bending pushes a single proton from the inside of the cell to the outside. Many such pumps working at once build up a difference in proton concentration across the membrane. That difference is, in effect, a tiny battery. The cell can use that battery to make ATP, the universal energy currency of life. And that is how a Archean makes a living in a salt pond with no chlorophyll at all. The color of that bacteriorhodopsin is purple. Purple in the most direct physical sense. When you grow a colony of Archaea in a flask, the flask turns lavender. When you look at a salt pond on the high Atacama, where conditions favor these creatures, the water turns the same delicate purple. That is not paint, not pigment in the way we usually imagine. That is a population of microbes drinking the green-yellow part of the sun and reflecting back the deep red and the violet and producing, by the simple physics of selective absorption, a soft purplish hue. So we know today that retinal-based photosynthesis exists. We know it works. We know it produces a purple color. And we know that the creatures that use it are some of the oldest, simplest, most ancient cell types still living on this planet. To answer that, we have to travel back. The purple Earth hypothesis asks past every how leaf, old exactly is this trick? Past every coral, past every fish, past every trilobite, past the first complex animals, past the first multicellular life, all the way back into a period of Earth's history that paleontologists call the Archean. The Archean eon ran roughly from 4 billion years ago to 2.5 billion years ago. It is the long, quiet stretch of our planet's youth. The Earth had finished its very first phase, the molten, glowing magma ocean stage of the Hadean. The crust had cooled. The first oceans had condensed out of the steamy atmosphere. The Moon, born of the Great Collision, had retreated to a still close orbit, looming much larger in the sky than today. The Sun was younger and dimmer by perhaps 30%. The atmosphere was thick with carbon dioxide and methane. There was almost no free oxygen anywhere. To breathe that air for even a moment would have killed any modern animal. And yet, on this strange young Earth, in those warm, low, methane-rich seas, life had taken hold. Microbial life, single cells drifting in the open water and clinging to rocks on the shallow seafloor. By 3.5 billion years ago, perhaps earlier, the planet was already alive in a quiet, tiny, invisible way. There were probably no large structures yet, no reefs, no layered mats of any great variety, just cells, slow, patient, biochemical experiments, repeating themselves in the warm water and on the wet rock. Some of these cells did not need the sun. They lived on chemistry. They drew their energy from hydrogen, from sulfur, from the warmth of hydrothermal vents on the deep sea floor. Their world was dark, and their food was the slow seep of minerals from the inside of the earth. They are the deep ancestors of organisms we still have today in the dark places of the ocean. Vent dwellers, sulfur eaters, the creatures of the inner earth, but others did need the sun. Or at least, others learned, somewhere in the long Archean, to take advantage of it. The sun was free. The sun was abundant. The sun was, even at 30% dimmer than today, an enormous flow of energy beating down on every surface of the shallow seas. Any cell that could find a way to use that energy would, over a very long time, gain a real advantage over the cells that could not. Such a cell would multiply more readily, it would spread further. It would, in the long arithmetic of evolution, become more common. The first organisms to drink the light, the Purple Earth hypothesis says, did so with retinal, not chlorophyll. Chlorophyll is a porphyrin-based molecule, more complex, requiring a longer biosynthetic pathway, requiring an iron or magnesium center, requiring more enzymes, requiring more genetic information. Retinal is simpler. It is a chain of carbon and hydrogen with a few oxygens, twisting under the touch of a photon. It is, in evolutionary terms, the cheaper, easier, faster solution. And the simple solution, in deep time, often comes first. There is a useful way to think about this, borrowed from how we think about technology in general. When a new function emerges in evolution, flight, vision, photosynthesis, anything, there are usually many possible biochemical paths to it. Some paths are short. They require few new proteins, few new enzymes, few coordinated genetic changes. Others are long. They require dozens of separate steps, each one of which has to provide some advantage on its own in order to be selected and preserved. Long paths are not impossible to evolve. They simply take longer. They tend to emerge after the short paths have already been worn smooth by use. Retinal is, in this sense, a short path. It can be assembled by a cell from materials that are already widely available. It needs only a few enzymes. The protein that holds it in the cell membrane is also relatively simple. The whole machinery for a primitive retinal-based light pump may require only a handful of new genes to come together in the right order. By contrast, a chlorophyll-based system, with its electron transport chains and its water-splitting enzymes and its multiple photosystems, requires a much more elaborate piece of biochemical engineering. Once you have it, it is enormously powerful. But getting there is, in evolutionary time, much harder. So the order suggested by the purple Earth hypothesis is not surprising. The simpler thing arrived first, took advantage of the abundant sunlight, and held the field for an enormous stretch of time, while the more complex thing was slowly assembling itself in some quiet corner of the biosphere. When the complex thing finally appeared, it could begin to compete with the simpler thing. And the chemistry it brought with it, especially the splitting of water and the release of oxygen,
Now, why
would
purple?
eventually
Why not some
change
other
the
color?
rules of the game.
To
For
understand
everyone.
that,
We have to think for a moment about the physics of light absorption. Sunlight, when it reaches the surface of the Earth, is not a single color. It is a spread of colors, reds and oranges in the long wavelength end, blues and violets in the short wavelength end, greens and yellows in the middle. The sun's light, weighted by intensity, peaks somewhere around 550 nanometers of wavelength in the yellow-green region. That is the part of the spectrum the sun pours out most strongly. That is where there is the most energy to be drunk. Bacteria rhodopsin, the retinal-bearing protein in the haloarchaea, has its strongest absorption peak right there, around 568 nanometers, right in the brightest band of the sun. It drinks the green and yellow light and reflects the red and the blue, and the visual mix of those reflected colors on a planetary scale would have looked to any observer floating in space distinctly purplish, a pale violet, a mauve, a lavender wash, especially across the shallow seas where life was densest. Chlorophyll, by contrast, absorbs in the red and the blue, and it bounces back the green and yellow. That is exactly the opposite arrangement. The two pigments, retinal and chlorophyll, drink completely different parts of the spectrum. They are, in a sense, mirror images of each other in the way they harvest sunlight. Where retinal-based life leaves the deep red and blue untouched, chlorophyll-based life leaves the green-yellow untouched. Some scientists have argued that this near-perfect complementarity is more than a coincidence. They suggest that when chlorophyll-based life first evolved, the Earth was already crowded with retinal-based life that had taken the prime, brightest band of the sun for itself. The newcomer, chlorophyll, had to make do with the leftovers, red and blue, less intense, less efficient in raw photon terms, but still useful and, crucially, available. And so the green pigment became green, not because green was the optimal choice, but because the optimal choice was already taken. This is a striking idea. It suggests that the color of every leaf, every stem, every patch of moss, every blade of grass on the modern Earth is, in effect, an inheritance, a historical accident, a choice made in deep time by the second color of life on this planet, working around the first. The forests are green, not because green is best, but because purple was first. Whether or not that exact framing turns out to be true in detail, the underlying point is interesting. The color of a biosphere is not a free choice. It is the result of which chemistry happened to evolve, in what order, on what world. Different worlds with different histories would, in principle, settle on different palettes. A planet whose first photosynthesizers used a slightly different pigment, slightly tilted in absorption, would look slightly different from space. Maybe more red, maybe more yellow, maybe a deeper, more saturated purple than ours ever was. The color of life on a planet is, in effect, a fingerprint of the long particular path that life took there. Our path took it through purple, then through green. Other planets, if they have life, are surely on their own paths. There is also a question of what counts as purple in any case. The retinal-bearing microbes today, when grown in dense culture, can range from a deep magenta to a soft violet to a distinct lavender, depending on subtle factors. The exact protein variant, the salt concentration, the temperature, the angle of the light. So when we say the early Earth was purple, we should picture a range, a mosaic. Different shades in different basins on different shores, some patches almost rose-colored, some patches almost gray-violet, some patches a delicate dusty mauve. Not a single color, but a family of related colors, all of them in the same gentle band. The way the modern biosphere, when seen up close, is not a single green, but 10,000 what greens. The Earth might have looked like in this purple period. Try to picture it slowly, the way you might picture a place from a story you are about to fall asleep inside. The sun is smaller, dimmer, about 3/4 as bright as today. It rises faster because the day is shorter, perhaps 18 or 19 hours long, the planet still rotating quickly under the slow tug of an early moon. The moon itself is larger in the sky than now, closer, brighter. A constant pale companion, looming overhead and influencing the rhythm of vast tides. The continents, such as they are not the continents you know. There may be only one large landmass, or a few small ones, depending on the exact age. The rock is bare. There's no soil in the modern sense, no grass, no animals, no insects. Wind and rain, working on bare basalt and granite, scour the surface and wash mineral-rich water into the seas. The oceans are warm, perhaps 50 degrees Celsius in places, and they smell of iron because the seas are full of dissolved iron in a form that today's oxygen-rich oceans cannot maintain. They smell of sulfur. They are dark and shallow and quiet. There are no fish. There are no jellyfish. There are no crabs and no clams and no coral and no kelp. There are only the smallest things, the cells, the films, the slow biochemistry of a living planet still finding itself. But across the surface of those warm seas, a great mat is forming, a thin, planet-wide skin of microbial life. In the most heavily populated regions, especially in shallow tidal zones and along sun-drenched coasts, the color of that mat is purple. Not bright, not vivid. A pale, soft, dusty violet, like the inside of a clamshell, or the color the sky takes on at the very last moment before night. From space, the seas and the wettest edges of the rock are tinted with this faint hue. Not uniformly. There are darker patches where life is dense, lighter patches where the rock shows through. A whole geography of pale lavender against gray stone. The sky above is not blue. It is yellowish, perhaps, or hazy and orange, depending on the methane content of the atmosphere and the dust kicked up by tides and weather. The sun's light, when it falls on the purple mats below, is mostly drunk. The protons pumped, the cells fed, the energy harvested. Whatever spills back, in the deep red and the soft violet, is what an external observer would see. That is the world. Not a green world, not yet a blue and green world, a world of warm, dim seas, lavender shallows, gray rock, and a young sun. For 2 billion years, give or take, that may well have been the dominant color of life on Earth. 2 billion years. Roughly half the age of the planet, far longer than the entire history of plants and animals combined. The Earth was, by this story, lavender for longer than it has been anything else. It is hard sometimes to feel the size of a stretch of time like that. 2 billion years is unimaginable. To get a sense of the proportions, consider this. If you compress the entire age of the Earth into a single calendar year, the formation of the planet falls on the 1st of January, and tonight falls on the last second of the 31st of December. Plants colonized the land somewhere in late November. The dinosaurs go extinct on the morning of the 30th. Recorded human history is the last 14 seconds before midnight. By that compressed scale, the purple Earth lasts from sometime in early February until sometime in early August. A whole spring and a whole half summer. The single longest, single-colored stretch in the planet's whole biological year. When we walk around our world today and assume that life is green, that life has always been green, that of course life is green, we are remembering only the smaller, later stretch. We are remembering autumn and winter in this calendar metaphor. The much longer spring, the high summer of the Archean, was not green at all. It was the long lavender hush of microbes drinking the brightest band of a younger sun in seas we would not entirely recognize, under a moon much closer than the one we know. There is something restful about realizing that. Our default sense of what the Earth looks like is recent. The world has worn other colors longer than it has worn this one. And it is all right, somehow, that this is so. The Earth has been many things, and our particular green and blue version is a young chapter, and one day there will be other chapters, and they too will pass, and the planet will keep on doing whatever it does very slowly, with great patience, in colors we have not yet seen, but of course it could not last forever. Somewhere in that long, slow purple stretch, another kind of cell was already emerging. Not in opposition to the purple cells exactly, just somewhere alongside them, a new kind of light harvester, one that used a more complicated molecule, that did its chemistry differently, and that almost by accident was about to change the entire planet beyond recognition. These were the Cyanobacteria, sometimes still called blue-green algae, although they are not algae and they are not technically blue. They are microbes, bacteria properly, and they had a different trick. They used chlorophyll. They had stitched together a more complex set of pigments and proteins, drawing on the red and blue ends of the spectrum, and they had also done something extraordinary. They had learned to split water. This is, on the face of it, a small chemical detail, but it changed everything. Older photosynthesis, the retinal kind, did not split water. It pumped protons, made ATP, and produced no oxygen as a byproduct. Cyanobacteria, by contrast, used the energy of light to break apart water molecules, taking the hydrogen for their own metabolism and releasing the oxygen as waste, free oxygen, a reactive, hungry, oxidizing gas into an atmosphere and an ocean that had never seen it before. For a long time, nothing visible happened. The oxygen the cyanobacteria released was almost immediately consumed. It reacted with iron in the seawater. It reacted with sulfur. It reacted with methane in the air. It bonded almost as fast as it was made with whatever it found and was locked away into rusts and sulfates and other minerals. The seas slowly turned. Iron precipitated out in vast layered deposits called the banded iron formations, which we still mine today as some of our richest sources of iron ore. Whole shallow basins over millions and millions of years became striped with red and dark gray, layer after layer, as iron and oxygen met and fell to the bottom. Then, around 2.5 billion years ago, a tipping point. The available iron in the oceans was largely used up. The available methane in the atmosphere was largely used up. The cyanobacteria kept making oxygen anyway. And for the first time in the history of the Earth, free oxygen began to accumulate in the air, quickly by geological standards. Within a few 100 million years, the atmosphere went from being almost free of oxygen to having a meaningful fraction of it. We call this transition the Great Oxidation Event. For most of the existing biosphere, this was a poison. Many of the older microbes, including many of the retinal-using ones, could not tolerate free oxygen. It tore at their membranes. It interfered with their chemistry. Some of them died out entirely. Others retreated into the few remaining places where oxygen could not reach. The deep mud, the salt-saturated brines, the dark anaerobic corners of the seafloor. The dominant color of the surface of the Earth, slowly, over hundreds of millions of years, began to shift, less purple, more green. The cyanobacteria, with their oxygen-tolerant biochemistry and their chlorophyll, spread outward. The retinal-based mats receded. By 1 billion or so years ago, the visible biosphere of the Earth was, almost certainly, dominated by the green pigment. The age of purple was over, but its survivors did not vanish. It is worth saying that this transition was, by the standards of human time, slow beyond imagining. We sometimes speak of the Great Oxidation as if it were an event, a moment, a boundary line on a chart, but it lasted, by some accounts, 200 million years from beginning to settled end. 200 million years, longer than the entire age of the dinosaurs, longer than the time since the rise of mammals. During every single one of those years, somewhere on the planet, the last large purple mats were thinning, and the first thick green mats were thickening, and the air was almost imperceptibly changing, and no observer of any year would have noticed any difference at all. The color shift that, in textbook drawings, looks like a sudden swap of paint was in lived time the patient grinding of geology and biology together, slower than any sense of now that any creature could possibly hold. There is also evidence that the climate suffered during this transition. The methane that had warmed the early atmosphere was being attacked by oxygen and broken down. As the methane fell, the greenhouse effect weakened, and the Earth, even with its slowly brightening sun, cooled significantly. There were, around 2.3 billion years ago, profound glaciations. Some scientists believe these were among the most severe ice ages in the history of the planet, possibly turning the entire surface briefly into a sheet of ice. The transition from purple to green was not a peaceful handover. It was, in slow geological motion, one of the more violent passages our planet has ever lived through. And yet, viewed from any one century inside it, nothing visible was happening at all. Just microbes
Today,
doing their
retinal-based
phototrophy is still the same as they had the year
It is,
before.
in fact,
more widespread than was understood until recently. We used to think of bacteria or rhodopsin as a curiosity confined to a few salt-loving microbes. Then, in the early 2000s, marine biologists began sequencing seawater for stretches of DNA at random, and they found that retinal-based light-harvesting genes, sometimes called proteorhodopsins, were almost everywhere in open ocean bacteria, in freshwater microbes, in dust-borne organisms, in archaea, in bacteria scattered across the tree of life. The proteins differ in detail, but the fundamental trick is the same. A retinal molecule sitting in a protein in the cell membrane, bending under the touch of a photon, pumping protons across, building up a tiny battery to run the cell. It is, in the modern ocean, a very common way to supplement an organism's energy budget. Many microbes today get only part of their living from light through these retinal proteins, and the rest from organic chemistry. But the light contribution is real, and it is global. The most visibly purple modern descendants are the Haloarchaea. They live in places almost no one else can. The salt pans of South America, the Dead Sea, hypersaline lakes on the high Tibetan Plateau, industrial salt evaporation ponds along the coast of California and the south of France. In all these places where the salt concentration is so high that almost any other living thing would dehydrate and die, Haloarchaea thrive. They have evolved exquisite ways to manage the osmotic pressure. They keep their internal salt concentrations even higher than their environment, and they pump protons with their bacteria or hodopsin, drink the light, and produce the color we see when we glance at one of those salty pools. If you fly over the salt evaporation ponds of San Francisco Bay in the right season, you will see a patchwork of pinks and reds and lavender purples, depending on which microbes dominate which pond. Some of those colors come from algae, some come from photosynthetic bacteria. But the deepest, most violet patches are typically the Haloarchaea. They are tiny living museums of the older world, cells whose biochemistry, in its essentials, has changed very little in 2.5 or 3 billion years. When you look at one of those purple ponds from the window of a passing airplane, you are, in a small way, looking at an Earth that was. There is a particular salt flat in the Atacama desert of Chile, very high, very dry, where conditions are unusually like those some scientists imagine for parts of the Archean Earth. Few clouds, strong sun, mineral-laden, salty water. Researchers have gone there specifically to look at the microbial communities, and they have found rich populations of retinal-using organisms, persistent, abundant, comfortable, in a chemistry that is not so different from ancient Earth's. It is one of the strongest modern arguments for the purple Earth hypothesis. The conditions still produce that biology where the conditions allow. If you stood at the edge of one of these high Atacama ponds in the cool morning before the sun had fully risen over the eastern range, you would see something very simple and very strange. The pond is shallow, a few centimeters deep perhaps. Its bottom is white with crystallized salt. The water above the salt is clear, but tinted faintly with violet. There are no fish. There are no insects skating on the surface. There are no water birds. There is only the wind and the slow drying of moisture into the dry air and the faint color of the water itself. That color is alive. The water is alive. Each drop of it contains millions of single-celled organisms, each of them carrying retinal in its membranes, each of them quietly pumping protons, drinking the early light. They are not trying to do anything. They are not aiming to be a color. They are simply running the same biochemistry they have been running, more or less unchanged, since long before any animal first left a footprint on the earth. You could stand there and not be able to tell by eye that the water is doing anything at all. There is no movement. There is no sound. There is no visible activity. Just the slow brightening of the morning and the stillness and the soft mauve tone of an old chemistry going about its business in a very thin, very salty, very ancient kind of way. In a sense, every salt pond like this is a small open window onto the Archean. Not a perfect window. The atmosphere is wrong. The sun is brighter than it was. The world around the pond is full of plants and animals and human voices that the early Earth never knew. But the chemistry inside the pond, the color of the water, the slow rhythms of the microbes, these things are old. Older than any animal, older than any cell with a nucleus, older than nearly anything on this planet that has not been preserved in stone. They are the oldest living thing you can look at without a microscope, and they are, perhaps, the closest thing that an ordinary human eye on an ordinary morning. There is one more place this story leads, and it is perhaps the most beautiful turn of all. If retinal-based photosynthesis was indeed the first widespread life pigment on Earth, and if that is because retinal is simpler and easier to evolve than chlorophyll, then it is reasonable to expect the same thing to have happened on other planets. Wherever there is a young world, with light and water, where life finds the same kind of biochemical opportunity, The simpler solution may emerge first there too. And if we ever look at such a world, with telescopes powerful enough to read its color, we may see something other than green. This is one of the ideas behind a research direction called exoplanet biosignatures. The pigment of life leaves a fingerprint in the way a planet reflects starlight. A green planet, dominated by chlorophyll, produces a particular small dip in the reflected spectrum in the red region called the red edge. It is a known target for astrobiologists looking for life beyond Earth. But if some planets are dominated by retinal-using life, instead of chlorophyll-using life, their spectral fingerprint will be different. They will absorb in the green-yellow band. They will reflect more at the red and blue ends. They will, from far away, appear purplish. So when the next generation of telescopes turns its attention to the rocky worlds around nearby stars, they will be looking, among other things, for what we might call a violet edge, a possible signature of an alien biosphere that has gone the way our own world apparently once did. A purple planet, somewhere out there, in some distant system, around some other star, doing what our planet did 3 billion years ago. Drinking the brightest band of its sun, reflecting the rest and quietly painting itself, by the unhurried physics of selective absorption, the color of an older life. We do not know how often that has happened in the universe. We do not know how often a young world chooses purple first, then later goes green, the way ours did. We do not know how often a young world stays purple. We do not know how often the experiment is interrupted by an asteroid, by a too active sun, by some catastrophe we have not yet imagined. We are at the very beginning of being able to look. But the color of life on other worlds is one of the few things we may someday be able to measure across light years. And in that question, somewhere, is the memory of our own planet. It is worth pausing at the end biology on what the purple Earth hypothesis is actually saying beyond the color itself. It is not saying that the early Earth was uniformly mauve. It is not saying that no other life existed. It is not saying that chlorophyll did not exist at all, or that cyanobacteria came late. The picture is more nuanced than that. The earliest photosynthetic life forms may well have been a mosaic, a long stretch of overlapping experiments. Different microbes in different places using different light-harvesting molecules, trying out the long, slow arithmetic of evolution. Some patches purple, some patches greenish, some patches gray or rust-colored. What the hypothesis is saying is that, on average, in the busiest sunlit places of the Archean Earth, retinal-based photosynthesis came first and dominated for a long time. That the simplest, fastest to evolve light harvester was used by the earliest sunlight-drinking life, that the green pigment, when it eventually emerged, found its niche around the edges of an already thriving purple biology, and that the changeover from one to the other was not a sudden replacement, but a slow shift over hundreds of millions of years in which oxygen rose, atmospheric chemistry transformed, and the dominant color of the visible biosphere quietly changed. There is something gentle about this picture. The Earth did not switch overnight. It did not lose its purple in an afternoon. The change was so slow that no individual cell would have known any difference at all. A generation of microbes, each living their few hours or few days, would simply have lived in the same pond their parents lived in, and made the same proteins and pumped the same protons. The transformation was at the level of the planet across geological time, working on a scale longer than any individual could measure. And the transformation has not entirely happened. Even now, billions of years later, the older biology has not been erased. It has been pushed into the corners, into the salt pans, the brine ponds, the high deserts, the deep dark mats below the oxygen line, into the places too harsh for the green descendants to follow. The age of purple is over, but it is not ended. It has retreated. It is quieter, smaller, narrower, and still very much alive. If, on a clear night, you could fly slowly over the right kind of salt pond, somewhere on the dry edge of a continent, and look down at the water in moonlight, you would see that lavender shimmer. You would see a thin film of older biology going about its ancient work, drinking light, pumping protons, carrying retinal in its membranes, the same molecule that once may have given the whole world its color, the same molecule that, in a slightly different form, is at this very moment registering these words in your eyes. So tonight, when you go to sleep, somewhere on this planet, in some warm, salty pond at the dry edge of a desert, the color of an older Earth is still showing, faint, patient, 3 billion years of memory in a thin film of microbes lying very still under a slow moon. The world used to be purple almost everywhere. Now it is purple only here and there. But here and there is enough. There is, finally, a kind of comfort in the long story of the color of life. We tend to think of our planet as a fixed thing, the blue and green ball of the photographs from space, the forests and oceans we recognize, the continents in their familiar shapes. But that fixed thing is, in deep time, an illusion of a single human moment. Continents move, oceans drain and refill, atmospheres rebuild themselves, pigments change, whole biospheres come and go. The Earth is not a single picture. It is a long, slow, patient sequence of pictures, each one beautiful in its own way, each one giving way to the next over millions of years, almost without anyone noticing. The purple Earth was one such picture. It lasted a long time. It was inhabited by life that was simpler than ours, more patient than ours, in some ways more durable than ours. Its descendants are still alive. Some of them are inside salt ponds, some of them are inside the open ocean. And one of them, retinal, the molecule itself, is inside almost every animal on the planet, doing a different job now at the back of every animal eye that has ever looked up at the night sky and wondered. When you close your eyes tonight, retinal and the photoreceptors at the back of your retinas will quiet down. The bending of those tiny molecules in response to the last fading light in your room will slow and eventually stop. Sleep is, in a small way, a withdrawal of light from the molecules that once, on a younger Earth, drank that light at scale. Your eyes, in a sense, are little inheritors of the older biology, a different use of the same chemistry, a new application of a very old trick. So when you drift off, you can think, if you like, of the color the world used to be, of the soft lavender shallows of an Archean coast, of the ancient mats of microbes 3 billion years ago, going quietly about their business under a smaller, dimmer sun, of the slow generations of cells, each one inheriting from the last, each one passing on the simple tools, retinal, the protein, the membrane, the proton pump, that would eventually, after enormous stretches of time, find their way into something as new and unlikely as your own ability to see. Sleep well. The Earth has worn many colors. It will wear more. And the older ones do not entirely vanish. They wait in quiet places in soft light for someone to remember them.